dear students of the Presidency University, members of the faculty of the university, and the leader, my dear friend, Professor Anuradha Lohia. Thank you very much for this wonderful New Year's gift. I've never imagined that I will speak and stand in the podium of a university which is 200 years old and help it march forward to infinity, because 2 times infinity is still infinity. So that's what it says, right? It's 200, but it's actually 2 times infinity. That's what I read it as. So thank you very much for this New Year's gift. Like the title of my talk suggests, I'm going to talk about cracking of the walls of classrooms. I've been a teacher for, I don't know, maybe 30 years, maybe 35 years, for quite a substantial period of time. And teachers are supposed to preach, and I'm not going to try to be preachy. So I'm going to start with an apology. And the apology is because my dear friend and the organizers of this program asked me for an abstract of my talk. And I went back, as I was trying to prepare this particular talk, I went back to my abstract and found that it was completely preachy. And so what I decided is that I'll apologize to you and just read my abstract for the first 30 seconds or so, and not even try to elaborate on the various sentences, because if I try to do so, I'll do exactly what I do not want to do, which is to preach. I do not want to be preachy today. I'm just going to give you a few examples to substantiate the remark that I made, or the claim that I've made, that the future appears bright because the walls of the classrooms are cracking. What's my apology? So that's what I'm going to read through, a few statements. The purpose of higher education is manifold. Education, especially higher education, is to instill in young people preparedness, good citizenship values, and competitive skills. Preparedness to think logically, preparedness to challenge dogma, preparedness to gain multiple domains of knowledge to serve the society. Values: to be a good human being, a good family member, and a compassionate and empathic community member. Skills: to perform professionally better than others competitively, and to work towards the improvement of society. This is what I believe comprises education. While skill sets are specific and need to be acquired for providing high-quality professional service to the society, for problem solving it is necessary to transcend from the specific to the general. In high schools, colleges, and universities, education is being imparted essentially to provide specialized skill sets to students. Often the mind is not prepared to think logically to solve a problem even after completion of a master's degree, and that's our failure. That's a failure of the classroom. Values of good citizenship are becoming rare elements. All this because I believe there is too much of specialized teaching that hardly provides any exposure to other domains of knowledge. Now here is a student who asks, I wish to become a biologist, why should I learn mathematics? And we hear this all the time. A student picks up biology, goes into higher education in biology because the student fears mathematics. And what I'm going to tell you today is some of the best biological research is done, it cannot be done without the appropriate mathematical skills, and I'll give you a few examples of that. A teacher says, why should we allow a biology student to take a course in statistics? And what I'm going to tell you today is to give you an example that some of the best biology is done by people who have skill sets in statistics. They also must know biology, they must ask the right questions in biology, but without the skill sets of statistics, those questions cannot be answered. I personally received education in an institution, and I met my founder twice as I came along to the De Rosio Hall. Prashant Chandra Mahalanobis, whose picture you see, who is graduated from the Presidency University, was a member of the 1912 batch, and he of course founded the Indian Statistical Institute, and that's the institute where I received my education, in which, as our founder envisioned, the walls of the classrooms were already cracked. I received my education, and I mean this metaphorically. They were not physically cracked. The building was not so old as it is today. Our institute is also many, many years old, not as old as the Presidency University. The reason why I say that I received education where metaphorically speaking, the walls of the classrooms were already cracked is because we, of course, had to learn mathematics and statistics. As I joined my Bachelor's of Statistics, I graduated from the Indian Statistical Institute with Bachelor's of Statistics, Master's of Statistics, and a Doctor of Philosophy in Statistics from the same institution. When we joined our BStat Honors program, we not only had to learn, of course we had to learn, that was the mainstay, mathematics and statistics, but we also had a fair mix of physical sciences, biological sciences, and social sciences, and all of us had to take that complete mix. There was no specialization so far as that part is concerned, and of course, we had to go through the grind of mathematics and statistics. So we had a kind of holistic education, and that's what I mean by saying that the walls of the classrooms were already cracked when I received my education. Today, even today, students in the Indian Statistical Institute who go through the BStat and the MStat programs, they get courses such as this: economic and official statistics and demography, geology, molecular biology, agricultural science, physiology, anthropology, sociology, physics, statistical methods in genetics, which is something that I started when I became a teacher or a member of the faculty in the institute, and that still continues. The only difference between the time from when I received my education at the ISI and today is that the walls of the classrooms, instead of cracking further, has actually come together, and that's very sad for me to see. The reason why the cracks, instead of becoming wider, have become closer is because today the students have to take these courses, but they have to choose one of the three streams: physical science, biological science, or social science. So you get specialized. You not only learn your mathematics and statistics, but instead of going through the whole, getting an exposure to the whole discipline of science, both social, physical, and biological, you now have to choose early on in which particular stream you want to get into. So I think that is less cracked than before, but still cracked. And that I think is fortunate, because the Indian Statistical Institute has not completely given up and said that we'll just do mathematics and statistics. Our founder believed that statistics cannot thrive without being exposed. Statisticians cannot thrive without being exposed to other kinds of sciences, and that's the reason why he had this, the holistic kind of a training program envisioned and put in place. But today, many of the teachers who sort of revised the courses felt that it was difficult to continue, and so they have this, specialization into one of these three sub-disciplines of science. Increasingly, teachers and students have realized that knowledge of multiple domains prepares one to be a better problem solver and be a better contributor to society. And that's what I personally believe, and I will give you examples as to why I believe so, and why I want to justify the subtitle of my talk, that the future is bright. Interdisciplinarity or multidisciplinarity is really integration of several disciplines to create a unified outcome. So you have a specific question, you want to see a specific outcome, and that particular outcome is only achievable, or oftentimes is only achievable by an integration of several domains of knowledge. And this is what I'm going to do for the rest of my talk. We began with reductionists, but now increasingly I don't have a pointer, unfortunately, otherwise I would have pointed to you, pointed out certain specific portions of my slides even as I go along, but I hope that it will become clear from my slides. We were all reductionists because we believed that reductionism helped us understand subsets of questions that were more focused, and we were able to get proper answers to those subsets of questions. But increasingly so, what we have learned to believe and learned to appreciate is that reductionism has its own properties and has its own restrictions, and therefore there is more and more integrative science that's beginning to emerge all across the various domains of knowledge. So I'm going to give you three examples, and may I request Professor Roy to give me a warning 5 minutes before, because I don't have really to complete all the three examples, but I'll try and do so. Hopefully I'll be able to relate to all of you because these are specialized examples, and there will be certain kinds of things that I will hand wave. Okay, so these are specialized, but I hope that I'll be able to relate to all of you, and I'm not going to explain every single detail except to give you a flavor that certain kinds of things could not be answered if one restricted oneself to one domain of knowledge. So we're talking about certain diseases, and when we talk about certain diseases, most of these diseases, or at least many of these diseases, run in families, and there are genetic faults that we try to discover. The diseases are due to fault in the gene, and the gene resides in the nucleic acid, which is called DNA. And we try and figure out where these faults are, because that gives us an empowerment to predict who might be predisposed to a specific kind of a disease. In cancers, most of these faults, and I'm going to begin my talk with one kind of cancer. In cancers, most of these faults happen during the life course of the patient. It's not inherited. So on the one hand, we say cancer is a fully genetic disease. On the other hand, cancer is really not heritable. It doesn't run in families. What happens is that we expose ourselves to various kinds of environmental carcinogens, such as tobacco smoke, such as the most potent biological carcinogen, human papilloma virus, and so on and so forth. So these carcinogens, they make changes in the DNA, and accumulation of these DNA changes eventually gives rise to cancer. But it only happens in that individual because it is not happening in what are called germ cells, that's transmitted from one generation to another. That happens in only about 10% of the patients. In the remaining 90% of the patients, it happens de novo, it happens only in that particular individual, and is not transmitted from one generation to another. So like I said, even though these changes are not heritable, cancer is really a disease of the genome because the changes are taking place in the DNA. So if you look at the evolution of our cancer genome, these are very nicely stacked cells, and certain kinds of changes take place as life goes on, and those dots are the cells in which changes are taking place. Most of these changes are innocuous. But sometimes one of these changes will give rise to a proliferation of that particular cell. So it's called a clonal increase or a clonal division of that particular cell. So that kind of division also mostly does no harm to the body. It's okay, except that change did give rise to a growth advantage to the cell, and that cell grew, made a small clone of itself. But those kinks and bumps are happening in us all the time, and we are completely oblivious to it, so we don't worry about those. Here is again another kink that has appeared because of a change in the DNA, but again completely innocuous. Then this red mutation or the red change happens, and this change is different from the other changes because this change gives rise to a huge growth advantage to the cell. The cell sort of crosses the epithelial barrier and becomes a malignant tumor. And so therefore, what we really need to understand are what are those cells or what are those DNA alterations in cells that gives rise to a huge growth advantage to the cell that it becomes a proliferative or malignant tumor. So we really need to identify those. It is a biological question, it's a biomedical question to be able to understand, to be able to identify those DNA changes that gives rise to a malignant tumor. And this is of course a major concern of all of humanity right now, cancer research is. So what we try and do, and of course there are other changes that take place in the background as well. So the question that we ask is, what are the genes and genomic alterations that drives this cell to having a great, wonderful growth advantage and becoming a malignant tumor? So that's what we need to identify. Why does this, so that's what we call as a driver or DNA alteration, and that's what we are really trying to identify. And it's not only one single driver, there are many drivers even when you're talking about cancer of a specific organ. This is impossible to, so what I'm going to try and impress upon you is that even though this is a fully biological or a fully genetic question, it is impossible to identify these drivers without statistical models and methods. And why do we need these statistical models and methods? What we do today is to use technology, and Mr. Vivek Kumar emphasized so much on usage of these technologies in education. We use what are called massively parallel or next generation DNA sequencing technologies, where I wish I had a pointer, where let's say that there are D and R, those are the two different types of DNA alterations that are there in the cell. And you see that there are many, if you look at that particular segment of the DNA and we have sequenced it multiple times, there are 2 blues appearing and there are a huge number of reds appearing. And what you see is that the original DNA strand, the thick bar, is really red. So all of these should really have been red, but sometimes you find these blues. And why do you find these blues? Because massively parallel sequencing platforms are error prone. Those blues are probably the small number of blues that we get in a large depth DNA sequence are probably errors, and those are because these sequencing platforms, these technologies are not completely error-free. There are potentialities of deriving errors. So what one needs is to be able to model that kind of an error, or to, first of all, to recognize that these platforms can have errors. You need to estimate what is the error. And why do you need to estimate that? Because if you didn't estimate that, then you would consider those 2 blue reads, these are called reads, those 2 blue reads as correct reads when in fact they're incorrect. So what you need to do is to calibrate what proportion of incorrect reads you might get in a massively parallel sequencing platform. Even if I'm going to skip the rest of it. I've made my point that one needs to recognize that these technology platforms are not error-free. One needs to be able to get estimates of these errors. Otherwise, you cannot calibrate your data against the errors, and you might consider the errors as signals when in fact they are not signals but only errors. So for that, okay, so that's one issue. The second issue is to be able to identify a driver. Like I said, DNA alterations are taking place in the genome all the time, but there are only these DNA alterations that gives rise, these few DNA alterations that give rise to the cell that get a huge growth advantage and become a malignant tumor. You need to identify that. To be able to identify a driver, first of all, we recognize that to be able to identify a driver, we need to calibrate against the background mutation rate. There are these changes that are taking place in the background, though that's really noise. We need to filter out the signals from the noise, and to be able to do that, we need to find those areas of the genome where the mutation rate is very high. Now, if you look at the entire genome, the mutation rate is not constant across the genome. The mutation rate is, the genome comprises important parts and less important parts. The less important parts, if the alterations happen, the body doesn't care, and therefore the mutations accumulate. But in the more important parts that code for certain kinds of proteins, if mutations take place, if DNA alterations take place, then protein changes happen, and the body doesn't usually, the individual doesn't usually tolerate those kinds of changes. The individual usually dies. So those are lethal mutations, and therefore those are not tolerated. So the main point that I'm making is that even the background noise level is not uniform across the entire length of the genome. And you need to recognize that and be able to estimate which part of the genome is evolving at what rate. Again, I'm going to skip this. We can't get a direct estimate of the mutation rate on the entire genome, so we fall back on other coordinates of the mutation rate, and we use those as surrogates in order to calibrate the background noise from the signal. And to be able to do that, I'm not trying to scare you, but all that I'm saying is that one needs models, and one needs models of an appropriate kind in order to be able to identify these driver mutations, and so we estimate background mutation rates in different parts of the genome and so on and so forth. I'll skip this as well. So essentially the point that I'm trying to make is that you need to be able to look at all of this, because without this kind of model and without these statistical estimates of the background rates and the error rates and so on and so forth, you would never ever be able to identify a driver. Using, of course, there are many ways of dealing with biology, using, of course, these kinds of DNA sequencing experiments. So we have been working on oral cancer, and oral cancer using all of these kinds of methodologies. What we have found is that there are certain genes, alterations in which leads to oral cancer, and these genes are predominantly tumor suppressors, as they are called, but don't worry about what tumor suppressors are. All that I'm saying is that this kind of work is done in India primarily because oral cancer is a very high relevance in India because of its high prevalence, especially among males, and that probably is a large fraction of that is probably attributable to the habit of chewing tobacco as opposed to smoking tobacco, which usually gives higher prevalence of lung cancer. when we look at these driver mutations, various driver alterations for oral cancer, we are also able to subclassify oral cancer. So we are able to show that all oral cancer is not a single homogeneous entity from a genetic point of view. You can actually use again statistical methods to subgroup, and this kind of tree making is also a statistical enterprise. But once you try and calibrate, once you try and classify the tumors into different kinds of, asking the question, what kind of DNA alterations led to this kind of tumor, what you find is that there are essentially three branches of this tree, and these three branches of this tree are predominantly three different kinds of genes that alterations in which lead to oral cancer. I'm going to skip this. Okay, so my first example was to impress upon you that we're asking a biological question. In asking this biological question, there are sub-questions such as error rates of the technology platforms, such as alteration rates across the entire genome that need to be modeled and taken into account in order to identify those driver mutations. So if your classroom walls are not cracked, if your teacher says, why should I allow my student of biology to learn statistics, it's a lost game. I'm going to talk a little bit about microbiology meeting human genetics. Oh, you can't see it? So anyway, one of the biggest things that has happened to humankind is the identification or the discovery or invention, however you want to look at it, of vaccines. And the impact of vaccines in the 20th and 21st century has been fantastic. Huge millions of lives have been saved because of these vaccines. Yet, when we, that's really a list of the various kinds of vaccines, various kinds of diseases, and the vaccines against those diseases, some of them have, the diseases actually, the infectious diseases have actually been eradicated completely, such as smallpox, such as polio, etc. And many other diseases have actually been contained to a large extent. So the impact of vaccines in 20th and 21st century has been phenomenal. Yet, a vaccine may not afford equal protection to all vaccinees. Not every person who is receiving the vaccine is protected equally well. And if you look at primary vaccine failure for measles, hepatitis B, and typhoid, typhoid is of special relevance to the city of Calcutta primarily because every year there is a typhoid outbreak in the slums in the Raja Bazar area and so on and so forth. And what you see is that 23 to 40% of vaccinees are not even protected against typhoid. So there is a poor protection for. The question is, why are these vaccinees not protected? In other words, why is there such a huge variation in the protection that's conferred by the vaccine to different individuals? Traditionally, the microbiologists have said that these pathogens, they mutate, they acquire DNA alterations, and therefore they get certain changes in their genomes because of which the same vaccine does not work against that pathogen. And therefore, if an individual, if an individual gets a vaccine which has a mutated pathogen, that particular vaccine will not work. That's one explanation, but that's not the only explanation. And the other explanation that many of us who do human genetics, who look at host-pathogen interactions, the host in this case is the human, the pathogen is the infectious agent. What we've been able to show is that, and when I say we, it's not us, not my laboratory, not our laboratory, but the global laboratory of science. When we talk about various other kinds of diseases such as measles, mumps, rubella, hepatitis, etc., what you see is that twin studies have shown that they have a very high association or correlation in terms of antibody response to vaccines. Fairly high. And this high correlation in antibody response really means that the twins, they actually share their entire genomes if you're looking at monozygotic twins. Of course, there's going to be an impact of the environment as well in terms of what kind of water the person drank before getting typhoid and so on. So these studies have shown that the heritability is high, which means that there may be a genetic component to antibody response to vaccines. So many of us ask this question, why is there so much of primary vaccine failure? And what we posited is that differences in genomic backgrounds of vaccine recipients not necessarily the changes in the pathogens, it's really the differences in the genomic background of the vaccine recipients that cause the primary vaccine failures. And of course, not all of us in the room will have exactly the same genome. No two of us will have exactly the same genome, even though we would share most of our genomes with each other, but still those small regions of the genome where we are different, those regions may play a role in conferring variability in vaccine response to the vaccinees. And so we did this study, a large study on typhoid vaccine. Typhoid is a major killer in parts of Calcutta. We did a major study in the Rajabaja slum area. We vaccinated individuals with informed consent. We provided them with the vaccine. We looked at them, on a time series basis, looked at their various immunoglobulins and so on and so forth, the components of vaccine response. And what we've been able to show is that there are certain genes that are located on these chromosomes that actually regulate, in some ways, the response to the typhoid vaccine. We've also done a similar study on cholera vaccine. Again, cholera is something that your Vice Chancellor is absolutely the expert on. But even so, what we've found is that it's not just the pathogen, it's also the genomic differences among the vaccinees that can regulate the impact of a particular vaccine and regulate the differences in response, in the primary response of the vaccine across the various vaccinees. So here is one where we look at a microbiological question, but we don't look at the pathogen per se. We also look at the host, we also look at the humans, recognize that the humans are different in terms of their genomes, and posit that these differences can play a role in the differences in primary vaccine response, and lo and behold, we are able to find regulatory changes in certain genes that are listed here in various chromosomes in various parts of the genome of the vaccine recipients. So this is another example of an interdisciplinary study where microbiology meets human genetics. My last example is statistics meeting computer science. I'm not a computer scientist, but over the years we've had to learn, many of us have had to learn some bit of computer science, primarily because the kind of data sets that we generate in order to answer the kinds of questions that we ask are so huge that standard statistical methods are not even working. So you can't use the standard statistical methods to analyze those kinds of data. So we fall back on certain ideas, certain algorithms that computer scientists have generated, and sort of blend the two in order to draw appropriate inferences from what are called big data. So there's a huge field that's emanating right now, not only in my own area of work, which is genetics, but also in other fields, such as physics, such as astronomy, where we generate huge quantities of data and don't know really how to analyze the data. What we had learned in statistics in our, in the education that we have obtained, those methods don't seem to work. We can't even handle those big data sets and draw appropriate inferences. So we fall back on computer science and other kinds of methods. And so let me just give you a flavor of the increase in data size per individual that we started generating. When I started my career, we were generating data that were about 2.5 kilobases long. And then about 10 years ago, we started generating genome-wide data, which were like 2 gigabases. And then these genetic data that we were generating comprised 50 points in the genome. When we were looking at genome-wide data, there were like 5 million points on the genome that we were assaying simultaneously. But now, and when we were generating genetic data, we were generating tens of individuals. When we started doing genome-wide data sets or generating genome-wide data sets, we were generating data on thousands of individuals, and this is per individual. So you can imagine the huge amount of data, increase in data volume and data size that was happening simultaneously. This is still the 1990s when we were generating data on kilobase scale. From 1990 onwards, we were generating data on gigabase scale, and now 2005 onwards, with the advent of the next generation sequencing machines, we are generating data on the terabyte or terabase scale. So there's been a huge increase in data volume. The place where statistics meets computer science is that is in order to fulfill this objective, that given the goal of inference, that you have a question, you have to answer that question, you have a particular inferential goal in mind, you discover a method of data analysis such that the quality of inference will be monotonical, quality of inference will monotonically increase with increase in data volume. This is your wish, that the more data that you generate, the better will be the quality of inference, but this usually does not happen because of our constraint of computation. Computational budget is highly constrained, and therefore the quality of inference does not necessarily monotonically increase with increase in data volume. So here's an example, and of course there are the computer scientists have envisioned or generated various kinds of tools such as machine learning tools that many algorithms for clustering, classification, regression analysis, and so on. The major problem with these machine learning kinds of tools is that the outputs of these algorithms don't provide error bars. In any inferential method, you need to not only provide an estimate, but you also need to provide an estimate of how confident you are about that estimate, right? So those are called error bars, and these algorithms usually don't provide that. And one of the things that was done about 20 years ago now, I think 25 maybe, is another technology called the bootstrap, where given one particular set of samples, we can't, the idea would be that if I measure n individuals, some generate data on n individuals, I can generate multiple sets of data on n individuals by taking another set of n individuals, a third set of n individuals, and so on, but that's going to be hugely expensive. Once I get, let's say, 3 or 5 sets of n individuals, I can also get 5 estimates, and therefore I can build an error of the estimate myself. But that's going to be hugely expensive because I'm going to have to bleed, let's say, 1,000 individuals 10 times. And first of all, getting consent of that many individuals is going to be difficult, and also bleed and generate data is also going to be very expensive. The best that we can accomplish is draw 1,000 individuals, get their informed consent, draw blood samples from them, generate data on massively massively parallel sequencing platforms, and using that, get an estimate, but also think of innovative ways of getting errors of those estimates. And that innovative way is called bootstrap, where we don't get multiple independent separate samples, but from the same data set we can resample and get other data sets that will be that are called bootstrap data sets that will provide estimates of the errors. So the bootstrap essentially then, you have one set of individuals x1 through xn, or one set of data x1 through xn, and you then resample and generate R different data sets from the same data, and you get different estimates from which you can get a global pooled estimate and also an error associated with it. So this is what Brad Efron in 1979 proposed, called the bootstrap, and that led the way to using merging computer science and statistics in some ways. But when the data sets became big, even this didn't seem to work, because if you're going to generate, resample from n, you really needed a huge computational power. And so what has been done more recently is by this is wrong, he's at UC Berkeley. Michael Jordan essentially proposed another method, which is merging of computer science ideas with statistics ideas, which is called a bag of little bootstraps. This might get a little bit technical, but essentially what it is that you don't resample the entire. So if you started with a sample size of n, you don't resample n anymore, you resample a subsample, which is m, and you draw m multiple times. But how do we guarantee that all this is going to work? So, so this is what's called the bag of little bootstraps. So you get little bootstrap estimates. How do we know that this is going to work? Then what you do is, if you, well, I don't have the theorem here. So he actually proved a theorem that if you draw samples which are smaller than your original sample of size n, you can draw smaller sample sizes and yet get a reasonably confident error estimate and an estimate of whatever you're trying to get, and an error associated with that estimate using this bag of little bootstraps. So essentially then, what I'm trying to say is that as our data sets are increasing and as we are unable to increase our computational budget, both in terms of hardware and also in terms of our ability to invest that kind of machine time, we have to get innovative ways of looking at big data sets, and this is one innovative way of looking at big data set that has merged both computer science ideas and ideas of statistics together in order to reach those inferential goals. Finally, the last two slides, challenges. The current system of academic advancement favors the specialized teacher or the independent investigator. So, like Mr. Vivek Kumar said in the beginning, that the Presidency University now has life sciences, a degree in life sciences, where students cannot get a job because they either have to be specialized in botany, zoology, microbiology, what have you. He has ensured that our students passing out of life sciences from Presidency University can get academic positions in universities in the state. But how many of the students can be supported only by universities in the state? We need a more national thinking about this, that these unified, integrated kinds of subjects need to be recognized and probably given more importance and prominence than specialized subjects such as botany, zoology, etc. Most institutions house teachers and scientists in discrete departments. So we have the we don't have a department of biological science, we have a department of botany, go to Calcutta University, every floor has a separate department, or two or three departments on the same floor. And more departments have proliferated even within the last 10 years in Calcutta University, and I can name some of those, and your Vice Chancellor is more cognizant of that or more familiar with that. Interdisciplinary science also requires interdisciplinary peer review, which is hard to come by. So there are occasions when many of us have faced that we've submitted a grant proposal for extramural funding, and competitive grant funding was talked about this morning by Mr. Vivek Kumar. We've submitted these kinds of interdisciplinary grant proposals for extramural funding when reviewers were not found. And as a matter of fact, again, I don't have to state my, in some ways, a very sorry beginning in this country, and then later, of course, it picked up. When her mentor, Professor Jyotirmoy Das, actually came to my rescue and recognized that it is important to blend statistics with genetics and fund such things extramurally. And he's no longer here, but I'm very grateful to him for having done that to me, and not to me, but to recognize this interdisciplinary science and to promote that. Not many of us are able to do that. Not many of us are even able to find peer reviewers to review the various kinds of project proposals that are being submitted. I would like to end with, again, a person who is very close to the city of Calcutta. Again, your Vice Chancellor knows her very well, Rita Caldwell. She works in the area of cholera. And this is what she said. She was the president of the National Science Foundation in the United States. And this is what, at some point of time, she said: we should always be looking for leaders who can foster interdisciplinary connections and thus advance scientific enterprise. I think this is very, very important. This leadership that recognizes the importance of interdisciplinary science, that recognizes the fact that walls of classrooms need to be cracked, is something that's absolutely crucial to the scientific enterprise. And on the occasion of the 200th anniversary of the Presidency University, I'm absolutely delighted to see that this university has started cracking the walls of classrooms, and therefore I think the future not only of Presidency University is bright, but of higher education in India is very bright because of the cracking of the classrooms. Walls of the classrooms. Thank you very much for your attention. Yeah, there was a question.
Thank you very much, Professor, for your exciting presentation. Though I never taught molecular biology or molecular medicine, but I get to answer these questions. We see a lot of money being spent on DNA sequencing and such kind of research all over the world. But we don't see a lot of money money spent on research on DNA, DNA editing.
DNA editing?
Gene editing.
Ah.
Gene editing. And also money on precision oncology. So what is the future of gene editing and future and precision oncology or precision medicine?
So precision, let me take the second question first. The precision oncology is in your clinics already, will be shortly in your bedroom. The impact of molecular biology, if molecular biology has made a major impact on any particular disease, invention of drugs, chemotherapeutic agents, management, it is cancer, in the area of cancer. Not all cancer types, but some cancer types, but still, the understanding is happening, and precision oncology will come. I have no doubt about that in my own mind. I could give a complete talk on oral cancer and how precision oncology or precision medicine can come about using these kinds of research. So the money is being well spent, I think. The second question about gene editing or genome editing, first of all, the technology is right now being perfected. The technology was invented, the technology is being perfected. If it is perfected, would we allow that to happen on ourselves is an ethical dilemma for which I do not have the answer right now. Some ethical dilemmas can be overcome. There are other ethical dilemmas that can never be overcome. We ourselves put a moratorium on certain kinds of dilemmas. I don't know what's going to happen to genome editing or gene editing per se, but right now what I would say is that a lot of money is being pumped in for perfecting the technology such that it becomes a little more precise or much more precise. Once it does, and we all hope that it will, but once it becomes very precise, what do we do with that technology is a completely different discussion that needs to take place. And like I said, maybe even a moratorium will be placed on genome editing. I don't know. Or maybe humankind will allow that to happen, modulo certain kinds of activities that may not actually be done with those kinds of technologies.